Hello, and welcome to another chapter of Advanced Pathophysiology. Today, in Chapter 1, we talk about the cellular biology. This is a bigger chapter, so I may be skipping over certain sections like what's new, some boxes, pictures. As always, please pause if you feel the need that you need to review something. I'll always give the opportunity to do that. So, without much further ado, I'll jump right in. All body functions depend on the integrity of cells. Therefore, an understanding of cellular biology is intrinsically necessary for an understanding of disease. An overwhelming amount of information is revealing how cells have a multicellular social organism. At the heart of cellular biology is cellular communication, how messages originate and are transmitted, received, interpreted, and used by the cell. This streamlined conversation between, among, and within cells maintains cellular function and specialization. Intercellular signals allow each cell to determine its position and specialized role. Cells must demonstrate a chemical fondness for other cells and their surrounding environment to maintain the integrity of the entire organism. When they no longer tolerate this fondness, the conversation breaks down and cells either adapt or become vulnerable to isolation, injury, disease, or even death. So, prokaryotes and eukaryotes. Living cells generally are divided into two major classes, eukaryotes and prokaryotes. The cells of higher animals and plants are eukaryotes, as are the single-celled organism fungi, protozoa, and most algae. Prokaryotes include cyanobacteria, bacteria, and rickettsia. Prokaryotes traditionally were studied as core subjects of molecular biology. Current emphasis is on the eukaryotic cell. Much of its structure and function has no counterpart in bacterial cells. Eukaryotes are larger and have more extensive intracellular anatomy and organization than do prokaryotes. Eukaryotic cells have a characteristic set of membrane-bound intracellular compartments called organelles that include a well-defined nucleus. Prokaryotes contain no organelles, and their nuclear material is not encased by a nuclear membrane. Prokaryotic cells are characterized by lack of a distinct nucleus. Besides having structural differences, prokaryotic and eukaryotic cells and chemical composition and biochemical activity. The nuclei of prokaryotic cells carry genetic information in a single circular chromosome, and they lack a class of proteins called histones, which in eukaryotic cells bind with deoxyribonucleic acid, or DNA, and are involved in the supercoiling of DNA. We now understand that the loops and coiling of DNA are important for many diseases. Eukaryotic cells have several chromosomes. Protein production, or synthesis, in the two classes of cells also differs because of major structural differences in RNA-protein complexes. Other distinctions include differences in mechanism of transport across the outer cellular membrane and differences in enzyme content. Cellular functions. Cells become specialized through the process of differentiation or maturation, so that some cells eventually perform one kind of function and other cells perform other functions. Cells with a highly developed function, such as movement, often lack other property, such as hormone production, which is highly developed in some other type of specialized cell. The eight chief cellular functions follow. Movement. Muscle cells can generate force that produce motion. Muscles that are attached to bones produce limb movements, whereas those that enclose hollow tubes or cavities move or empty content when they contract. An example, the contraction of smooth muscle cells surrounding blood vessels changes the diameter of the vessel. The contraction of muscles and walls of the urinary bladder expels urine. Conductivity. Conduction, as a response to a stimulus, is manifested by a wave of excitation, an electrical potential that passes along the surface of the cell to reach its other parts. Conductivity is the chief function of nerve cells. Metabolic absorption. All cells take in and use nutrients and other substances from their surroundings. Cells of the intestine and the kidney are specialized to carry out absorption. Cells of the kidney tubules reabsorb fluids and synthesize proteins. Intestinal epithelial cells reabsorb fluids and synthesize protein enzymes. Secretion. Certain cells, such as the mucus gland cells, that can synthesize new substances from substances they absorb and then secrete the new substances to serve as the needed elsewhere. Cells of the adrenal gland, testes, and ovary can secrete hormonal steroids. Excretion. All cells can rid themselves of waste products resulting from the metabolic breakdown of nutrients. Membrane-bound sacs within cells contain enzymes that break down or digest large molecules, turning them into waste products that are released from the cell. Respiration. Cells absorb oxygen, which is used to transform nutrients into energy in the form of adenosine triphosphate, or ATP. Cellular respiration or oxidation occurs in organelles called the mitochondria. Reproduction. Tissue growth occurs as cells enlarge and reproduce themselves. Even without growth, tissue maintenance requires that new cells can be produced to replace cells that are lost normally through cellular death. Not all cells are capable of continuous division. Communication. Communication is vital for cells to survive as a society of cells. Pancreatic cells, for instance, secrete and release insulin, necessary to signal muscle cells to absorb sugar from the blood for energy. Constant communication allows the maintenance of a dynamic, steady state. So the structure and function of cellular components. The following figure shows a typical eukaryotic cell. It consists of three components: an outer membrane, called the plasma membrane, or plasma lemma, a filling called cytoplasm, and the intracellular organs, or organelles, which are membrane-bound and include the nucleus. Researchers are astounded by the advances in microscopic and computer software that allows the resolution to the nanoscale. Cells seem to come alive with the molecular world more visible. Understanding structure and function will reveal, for example, how the cell responds to mechanical forces or emerges from different gene expression patterns. The overall impact on biology is huge. So here we're taking a look at a cell. The typical or composite cell. Again, hit pause if you need to do a refresher of your biology. The nucleus. The nucleus, which is surrounded by the cytoplasm and generally is located in the center of the cell, is the largest membrane-bound organelle. Two pliable membranes comprise the nuclear envelope. The nuclear envelope is pockmarked with pits called nuclear pores, in which nuclear pore complexes are positioned that allow molecules to move between the nucleus and the cytosol. The outer membrane is continuous with membranes of the endoplasmic reticulum. The inner membrane encloses the nucleus. The nucleus contains the nucleolus, a small, dense structure composed largely of RNA. Most of the cellular DNA and the DNA-binding proteins, the histones, that regulate its activity. The DNA chain in eukaryotic cells is so extensive that the risk of breakage is high. Therefore, the histones binding to DNA cause DNA to fold into chromosomes. The wrapping of DNA into tight packages of chromosomes is essential for cell division in eukaryotes. So here we're taking a look at the nucleus. Again, if you need a refresher for your biology, Here's a good spot to pause so you can review it. The primary function of the nucleus are cell division and control of genetic information. Other functions include the replication and repair of DNA and the transcription of the information stored in DNA. Genetic information is transcribed into RNA, which can be processed into messenger, transport, and ribosomal RNA, and introduced into the cytoplasm, where, where it directs cellular activities. Most of the processing of RNA occurs in the nucleolus. The cytoplasmic organelles. Cytoplasm is an aqueous solution that fills the cytoplasmic matrix, the space between the nuclear envelope and the plasma membrane. The cytosol represents about half of the volume of a eukaryotic cell. It contains thousands of enzymes involved in intermediate metabolism and is crowded with ribosomes that make proteins. The cytosol is the main site for protein synthesis and degradation. Newly synthesized proteins remain in the cytosol if they lack a sorting signal for transport to a cell organelle. The organelles suspended in the cytoplasm are enclosed in biologic membranes, which enable them to simultaneously carry out functions that require different biochemical environments. These functions, many of which are directed by coded messages carried from the nucleus by RNA, include synthesis of proteins and hormones, and the transport out of the cell, isolation and elimination of waste products from the cell, metabolic processes, breakdown and disposal of cellular debris and foreign proteins, and maintenance of cellular structure and motility. Also, the cytosol functions as a storage unit for fat, carbohydrates, and secretory vesicles. Ribosomes. Ribosomes are RNA protein complexes that are synthesized in the nucleolus and secreted into the cytoplasm through pores in the nuclear envelope, called nuclear pore complexes. These tiny ribosomes may float free in the cytoplasm or attach themselves to the outer membranes of the endoplasmic reticulum. Their chief function is to provide sites for cellular protein synthesis. Newly formed ribosomes synthesize a recognition sequence, or signal, like an address on a letter. Signal recognition particles in the cytosol bind to the ribosome after recognizing the SRP, the signal recognition particles. Ribophorins, receiver proteins found in the rough section of the endoplasmic reticulum, act as the address, site, or binding site. The developing protein threads its way through the endoplasmic reticulum membrane into the lumen. The signal recognition particle is removed and the new protein chain is folded into its final conformation. The endoplasmic reticulum. The endoplasmic reticulum is a membrane factory that specializes in the synthesis and transport of the protein and lipid components of most of the cell's organelles. It consists of a network of tubular or sac-like channels that extend through the cytoplasm and are continuous with the outer nuclear membrane. The folded membranes that comprise the cisternae of the endoplasmic reticulum may be rough or smooth. The rough endoplasmic reticulum is rough because ribosomes and ribonucleoprotein particles are attached to it. Some of the proteins synthesized by these ribosomes remain in the endoplasmic reticulum, and others are used to construct membranes of other organelles and of the cell itself. Importantly, the endoplasmic reticulum is responsible for much of the cell's protein synthesis and folding, and a new role of the endoplasmic reticulum is sensing cellular stress. Understanding mechanisms of cellular stress will aid diagnosis and treatment of disease. So here we're taking a look at a picture of endoplasmic reticulum. Again, hit the pause button if you need to do a small refresher of your biology in this aspect. We will be skipping over the What's New section. Again, if you want to read this, go ahead and hit pause. And I'll stop on several sections so you can read all of it. Smooth endoplasmic reticulum does not contain ribosomes or ribonucleoprotein particles. Rather, membranous surfaces of the smooth endoplasmic reticulum contain enzymes involved in the synthesis of steroid hormones and are responsible for a variety of reactions required to remove toxic substances from the cell. The endoplasmic reticulum communicates with the Golgi complex and interacts with other organelles, particularly lysosomes and peroxisomes. So what is the Golgi complex? The Golgi complex is a network of flattened, smooth membranes and vesicles frequently located near the nucleus of the cell. Proteins from the endoplasmic reticulum are processed and packaged into small membrane-bound sacs or vesicles called secretory vesicles, which collect at the end of the membranous folds of the Golgi bodies, called cisternae. The secretory vesicles then break off from the Golgi complex and migrate to a variety of intracellular and extracellular destinations, including the plasma membrane. The vesicles fuse with the plasma membrane, and their contents are released from the cell. The best known vesicles are those that have coats made largely of protein clathrin and are called clathrin-coated vesicles. They bud from the Golgi complex on the outward secretory pathway and from the plasma membrane on the inward endocytic pathway. Newly synthesized endoplasmic reticulum proteins are carried from the Golgi network to the endosomes by clathrin-coated transport vesicles before progressing to lysosomes. Many molecules, including lipids, proteins, glycoproteins, and enzymes of lysosomes, pass through the Golgi complex at some stage in their maturation. The Golgi complex is a refining plant and directs traffic in the cell. So here we're taking a look at the Golgi complex. Again, feel free to hit pause so you can kind of review what it looks like and how the system works in that sense. Again, here's a good spot to pause so you can review the cell structure and function and how the internal membrane system of the cell packages the protein for export. So that leads us to lysosomes. Lysosomes are membrane-enclosed organelles filled with enzymes that digest macromolecules and defunct intracellular organelles and particles engulfed from outside the cell by endocytosis. They contain more than 60 digestive enzymes called hydrolases, which catalyze bonds in proteins, lipids, nucleic acids, and carbohydrates. Hydrolases function optimally at an acidic pH. Lysosomes function as the intracellular digestive system. Lysosomal enzymes are capable of digesting most cellular constituents completely to their basic components, such as amino acids, nucleotides, and carbohydrates. Transport proteins in the lysosome membrane carry these final components to the cytosol, where the cell can reuse or excrete them. Recent data have radically shifted the view that lysosomes are not just trash cans and recycling agents, but have established a central role of the lysosome in nutrient-dependent signal transduction for cellular adaptation. The newly discovered signaling function cooperates with the known degradative role of the lysosome to mediate basic cellular function, such as nutrient sensing metabolic adaptation and quality control of proteins and organelles. Complex transcriptional programs control the synthesis, composition, and quantity of lysosome and regulate their activity to match the evolving needs of the cell. Alterations in these essential functions are central to the pathophysiology of an expanding range of conditions and include storage diseases, neurodegenerative diseases, and cancer. Lysosome maintains cellular health because of efficient removal of toxic cellular components, removal of useless organelles, termination of signal transduction, and maintenance of metabolic homeostasis. Aging can lead to progressive loss of lysosomal efficiency and decline of the regenerative capacity of organs and tissues. Lysosomes are key signaling hubs of the sophisticated network called cellular adaptation, and the network includes ion and nutrient transporters, protein kinases and phosphatases, and transcription factor and transcriptional regulators. Although these components integrate functions such as nutrient abundance, energy levels, and cell stressors, and translate them into instructions that regulate cellular metabolism toward either proliferation or inactivity, the signaling function have a far-reaching implication for metabolic regulation in health and in disease. So again, here's a good spot to hit pause so you can take a look at how the cells function with their lysosomes. So here you have the endolysosomal system and four pathways to degradation in lysosomes. So if you look at picture A and B, you'll review that. The lysosomal membrane acts as a protective shield between the powerful digestive enzymes within the lysosome and the cytoplasm, preventing their leakage into the cytoplasmic matrix. Disruption of the membrane by various treatments or cellular injury leads to the release of the lysosomal enzyme, which can then react with their specific substrates, causing cellular self-digestion. Lysosomal abnormalities are involved in a number of conditions that involve cellular injury and death. Lysosomal storage diseases may be the result of a genetic defect or lack one or more lysosomal enzyme. For example, the lack of lysosomal alpha 1 4-glucosidase leads to an accumulation of glycogen in lysosomes known as Pompe disease. Tay-Sachs disease is characterized by an accumulation of a GM2 ganglioside in lysosomes as a result of the deficiency or absence of lysosomal hexosaminidase A. In gout, undigested uric acid accumulates within lysosomes, damaging the lysosomal membrane. Subsequent enzyme leakage results in cell death and tissue injury. Four pathways of degradation in lysosomal include endocytosis, phagocytosis, macropinocytosis, and autophagy. Endocytosis is the uptake of the macromolecules from extracellular fluid. Phagocytosis is engulfment of large particles or microorganisms in phagocytic cells, like macrophages and neutrophils. Macropinocytosis is the nonspecific uptake of fluids, membrane, and particles attached to the plasma membrane, and autophagy begins in the cytosol and is used to digest cytosol and ineffectual organelles. Extracellular substances are taken into the cell and encapsulated in the membrane-bound vesicles. Lysosomes merge with the vesicle to form a digestive vacuole. Lysosomes remain fully active by maintaining a lower internal pH. They do this by pumping hydrogen ions into their interiors. The hydrolytic enzymes are only maximally active at acidic pH values. Lysosomes that are not active do not maintain such an acidic internal pH. Lysosomes in this holding pattern are called primary lysosomes. When a primary lysosome fuses with a vacuole or other organelle, its pH decreases and the hydrolytic enzymes become activated. When it becomes active, it's called a secondary lysosome or heterophagosome. As cells complete their lifespan and die, lysosomes digest the resultant debris or obsolete parts by autophagy. Lysosomes involved in this process, which is called autodigestion, are called autolysosomes or autophagosomes. In living cells, cellular debris is encapsulated within the cell and vesicle that reacts with a lysosome to complete its degradation. Autophagy promotes homeostasis because it involves continuous biosynthesis and the cell turnover. Therefore, autophagy plays a crucial role in health. Defects in autophagy may challenge disposal mechanisms from removing microbes, unnecessary protein aggregates, and abnormal proteins, resulting in contributions to disease ranging from infectious disorders to neurodegeneration and cancer. Products of autophagy pass out of the lysosome and are reused by the cell. Indigestible material is stored in vesicles called residual bodies, whose contents are actively expelled from the cell. High concentrations of lipids may accumulate within the residual bodies and remain there for a long time. The lipids are eventually oxidized, and a pigmented substance containing polyunsaturated fatty acids and proteins accumulates in the cell. This pigmented substance, termed lipofuscin, is often called age pigment or age spots and is noted in older individuals. Peroxisomes. Peroxisomes are membrane-bound organelles that contain several oxidative enzymes, such as catalase and urate oxidase. These oxidative enzymes can detoxify compounds and fatty acids. Similar to lysosomes in microscopic appearance, peroxisomes are larger and oval or irregular in shape. Like mitochondria, peroxisomes are major sites of oxygen utilization. Peroxisomes are so named because they usually contain enzymes that use oxygen to remove hydrogen atoms from specific substrates in an oxidative reaction that produces hydrogen peroxide. Hydrogen peroxide is a powerful oxidant, potentially destructive if it accumulates or escapes from peroxisomes. Catalase, an antioxidant enzyme, uses the hydrogen peroxide to oxidize a variety of other substrates, your phenols, your formic acid, formaldehyde, and alcohol, by the peroxidative reaction. Thus, the breakdown of hydrogen peroxide yields water and oxygen. Peroxisomes also have important role in the synthesis of specialized phospholipids necessary for the nerve cell myelination. Such reactions are important in detoxifying various wastes within the cell or foreign components that enter the cell, such as ethanol. Impairment of peroxisomes can lead to disease. That brings us to the mitochondria. Mitochondria organelle found in large numbers in most cells are responsible for cellular respiration and energy production. These cytoplasmic organelles appear as spheres, rods, or filamentous bodies that are bound by a double membrane. The outer membrane is smooth and surrounds the mitochondria itself. The inner membrane is convoluted in the mitochondrial matrix to form partitions called cristae. The inner membrane contains the enzymes of the respiratory chain, the name given to the electron transport chain. These enzymes are essential to the process of oxidative phosphorylation that generates most of the cell's ATP. Metabolic pathways involved in the metabolism of carbohydrates, lipids, and amino acids, and special pathways involving urea and heme synthesis are located in the mitochondrial matrix. So here's a picture of a mitochondria, its inner and outer membrane. And here you take a look at it on a cellular view. The outer membrane is permeable to many substances, but the inner membrane is highly selective and contains many transmembranous transport systems. The inner membrane contains transporter to move electrically charged calcium. Mitochondria contain their own DNA that codes for enzymes needed for oxidative phosphorylation. Your cytosol. Cytosol is a gelatinous, semi-liquid portion of the cytoplasm, accounting for about 55% of the total cell's volume. Functions of the cytosol include intermediary metabolism involving enzymatic biochemical reactions, ribosomal protein synthesis, and storage of carbohydrates, fat, and secretory vesicles. Intermediate or intermediary metabolism refers to the intracellular chemical reaction that includes synthesis, degradation, and transformation of small organic molecules. All intermediary metabolism occurs in the cytoplasm, or that portion of the cell interior not occupied by the nucleus, with most of the metabolism being accomplished in the cytosol. These reactions enable energy to be used for managing cellular activities and for providing substrates to maintain cell integrity. Ribosomal protein synthesis takes place in free ribosomes in the cytosol. Cytosolic ribosomes that synthesize identical proteins are collected in factories known as polyribosomes. Excess stored nutrients not immediately used for ATP production are converted into cytosol into storage forms. For example, excess glucose is stored as glycogen. These temporary masses are known as inclusions. Secretory vesicles that have been processed and packaged by the endoplasmic reticulum and Golgi complex also remain in the cytosol. By means of signaling, the vesicles transport and empty their contents outside the cell. The cytoskeleton. All eukaryotic cells contain elaborate and specialized internal structures in the cytosol that provide the bones and muscles of the cell. The cytoskeleton. The cytoskeleton maintains the cell's shape and internal organization, and it permits movement of substances within the cell and movement of external projections outside the plasma membrane. The internal skeleton is composed of a network of protein filaments. The three main types of filaments include actin filaments, microtubules, and intermediate filaments. These filaments collectively promote cell strength, shape, and movement. By translating mechanical forces and deformations into biochemical signals, cells sense their physical environment, including the extracellular matrix, neighboring cells, and physical stress. The cytoskeleton is involved in the extracellular matrix and nuclear interior in force transmission, called mechanotransduction. Mechanotransduction describes the cellular processes that translate mechanical stimuli into biochemical signals, allowing cells to adapt to their surroundings. Cell stresses, however, that involve adaptations to mechanotransduction are associated with several alterations and diseases, including loss of hearing, cardiovascular disease, muscular dystrophy, and cancer. Microtubules are small, hollow, cylindrical, unbranched tubules made of protein. When found together, microtubules exhibit rigidity unlike the rest of the cytoplasm. Microtubules thus add strength to the cell structure. Within the cell's microtubules support and move organelles from one part of the cytoplasm to another, facilitate transport of impulses along nerve cells, and have roles in the inflammatory and immune responses and hormone secretion. Microtubules are also involved in the external movement or motility of some cells. So here you're taking a look at a picture of the cytoskeleton. Again, a good spot to pause if you need a biology refresher. Microtubules are arranged in the thickened base, or basal body, of a protrusion from the cell's plasma membrane. This arrangement occurs in the basal bodies of sperm flagella and the cilia of certain other cells. The long whip-like flagella enable the movement of sperm cells. Cilia usually move substances past the cell, which remains stationary. For example, cilia on cells lining the respiratory tract move together to beat mucus towards the throat so that it can be removed by coughing. While the cell is not in the process of division, only a few microtubules are assembled. Cellular division or defense does, however, induce a cycle of rapid assembly and disassembly. Microtubules involved in cellular division are arranged in a centriole. Centrioles always consist of 9 bundles, containing 3 microtubules each. During division, the pairs of centrioles split and migrate to opposite poles of the cell. Your actin filaments are smaller fibrils that generally occur in bundles rather than a single entity. They are concentrated in the cell cortex, just beneath the plasma membrane. The cortical actin network is a major driver for many cell functions, including cell movement, endocytosis, and maintenance of cell and tissue shape. Actin filaments link the interior of the cell to adjacent cells through cell junctions. In addition, microfilaments are necessary for regulating cell growth and drive the pinching of one cell into two. Cellular locomotion depends on contractile properties that involve both microtubules and actin filaments. The actin cytoskeleton in motion in motile cells are recently been described as a wave of excitation that may account for the spontaneous migration of cells. Intermediate filaments are braided, rope-like fibers made of several filament proteins. The different filaments form a mesh called the nuclear lamina beneath the inner nuclear membrane, creating a protective chamber for the cell's DNA. Other types crisscross the cytoplasm, promoting mechanical strength and epithelial tissue. These filaments bring the cytoplasm from one cell junction to another, supporting and strengthening the sheet of epithelium. So now we can talk about the plasma membranes. Membranes define the cell's boundaries, whether they surround the cell or enclose an intracellular organelle. Membranes are crucial to normal physiologic function because they control the composition of the space or compartment they enclose. They can allow or exclude various molecules, and because of selective transport systems, they can move molecules in or out of the space. By controlling the movement of substances from one compartment to another, membranes exert a powerful influence on metabolic pathways. Directional transport is facilitated by polarized domains, distinct apical and basolateral domains. The direction of cells, cell polarity, maintains normal cell and tissue structure for numerous functions. Most importantly, transport of nutrients in and out of the cell and become altered with disease such as cancer. In addition to these functions, the plasma membrane has an important role in cell-to-cell recognition. For example, protein receptors for hormones and for other chemical signals are associated with the membrane and act as markers that identify a cell to its neighbor. Other functions of the plasma membrane include assistance with cellular mobility and maintenance of cellular shape. So here we're taking a look at the transport channel of enzymes, cell surface receptors, cell surface markers, cell adhesion, and attachment of cytoskeletons. So the function of the plasma membrane, proteins. Here's a good spot to pause so you can kind of review how that membrane can differentiate what is going to come in and out of a cell. And here, the spot impression, you see the cell polarity of epithelial cells. Again, a good spot to pause to read the slide and kind of review it. Table 1.1, Plasma Membrane Functions. Again, if you're not too familiar with the biology, here's a good spot to pause, review the cellular mechanism of different membrane functions. So, membrane composition. Membrane composition. The main components of cell membranes are lipids and proteins. The basic structure of cell membranes is the lipid bilayer, composed of two continuous opposing leaflets and proteins that span the bilayer or interact with the lipids on either side of the two leaflets. The lipid bilayer provides the basic fluid structure of the membrane and is mostly an impermeable barrier to water-soluble molecules. Individual lipid molecules can diffuse readily throughout their own monolayer. Most membrane proteins span the lipid bilayer and mediate most of the functions of the membrane, including transport of molecules across the membrane and ATP synthesis. So here we're taking a look at your lipid bilayer and what can and cannot come in and out of that cell membrane. So here's a good spot to pause to review and take a look at what you're looking at. And I'll go down a little bit so you can also read along the lipid bilayer membrane. Again, a good spot to hit a pause button. And here's a full explanation of what you're looking at. So another good spot to pause. Historically, the plasma membrane was described as a fluid lipid bilayer composed of a uniform lipid distribution with inserted moving proteins, where the lipid molecules in the plasma membrane and living cell membranes segregate into domains called lipid rafts, is a controversial topic. It is known that many lipids and proteins are not distributed uniformly, but are seen to concentrate in a temporary dynamic way, assisted by protein-to-protein interactions that enable transient formation of domains or lipid rafts. Organization of lipid rafts in living cells may be important for cell communication, where protein assemblies convert extracellular signals into intracellular signals. Larger assemblies of clusters are cavolae, flask-shaped invaginations, thought to form from lipid rafts and to be important for endocytosis. Carbohydrates are mainly associated with plasma membranes, where they are chemically combined with lipids to form glycolipids and with proteins to form glycoproteins. So, what are lipids? Cell membranes may contain many different lipid classes, but in animals, the main ones are phospholipids, cholesterol, and glycolipids. The most abundant lipid are phospholipids. Phospholipids are key for repairing the membrane. They tend to spontaneously rearrange themselves to avoid a tear. By folding on themselves and forming a sealed compartment, inositol phospholipids are a subclass of phospholipids important for cell signaling because in the cytosolic lipid leaflet of the bilayer, they respond to extracellular signals. Lipids, along with protein assemblies, act as molecular glue, so the structural integrity of the membrane. Each lipid molecule is said to be polar, or amphipathic. An amphipathic molecule is one in which one part is hydrophobic and another part is hydrophilic. So one side loves water and one side hates water. The membrane spontaneously organizes itself into the bilayer because of these two incompatible solubilities. The hydrophobic region of each lipid molecule is protected from water, whereas the hydrophilic region is immersed in it. The bilayer structure accounts for one of the essential functions of the plasma membrane. It is impermeable to most water-soluble molecules because they are insoluble in the oily core region. The bilayer serves as a barrier to the diffusion of water and hydrophilic substances while allowing lipid-soluble molecules, such as oxygen and carbon dioxide, to diffuse through it readily. Because the bilayer is fluid at temperatures greater than freezing, components of the cellular environment move slowly and selectively across the membrane at all times. So here you're taking a look at the structure of the phospholipid molecule, where you see the hydrophilic head and the hydrophobic head. Proteins. Proteins perform most of the plasma membrane's specific tasks. The amount of types of proteins in the membrane vary. A protein is made from a chain of amino acids, known as polypeptides. There are 20 types of amino acids in proteins, and each type of protein has a unique sequence of amino acids. Proteins are the major workhorses of the cell. Membrane proteins associate with the lipid bilayer in different ways, including transmembrane proteins extending across the bilayer and exposed to an aqueous environment on both sides of it. Proteins located almost entirely in the cytosol and associated with the cytosolic half of the lipid bilayer by an alpha helix exposed on the surface of the protein. Proteins existing outside the bilayer on one side or the other and attached to the membrane by one or more covalently attached lipid group, and proteins bound indirectly to one or the other bilayer membrane, faced and held in place by their interactions with other proteins. Membrane proteins are amphiphilic, with both hydrophobic and hydrophilic regions. So here you're taking a look at the four different types of proteins and how they attach to the plasma membranes, whether they are, a transmembrane, monolayer associated with the alpha helix, a lipid-linked, or protein attached. Proteins exist in densely folded molecular configurations rather than straight chains, so an excess of hydrophilic units is at the surface of the molecule, and an excess of hydrophobic units is inside. Membrane proteins, like the proteins, are synthesized mainly by ribosomes in the cytosol and then travel, called trafficking. Their fate depends on their amino acid sequence, which contains sorting signals that direct their delivery to locations outside the cytosol or to organelle surfaces. A simplified illustration of protein traffic within a cell is presented in Figure 1.114. So here you see the simplified roadmap of the protein traffic. Again, a good spot to hit pause so you can see how your cytosol travels up and down throughout the cell. Proteins move from one compartment to another by gated transport, protein translocation, vesicular transport. In gated transport, selective gates direct the movement of proteins and RNA molecules between the cytosol and the nucleus through nuclear pore complexes in the nuclear envelope. In protein translocation, transmembrane protein translocators directly transport proteins across a membrane from the cytosol to a different, distinct location. To snake through the translocator, the protein molecule usually unfolds. In vesicular transport, the bubble-like vesicle is a membrane-enclosed intermediate of either a small transport vesicle or a large organelle fragment. These vesicles discharge their cargo into a different compartment by fusing with the compartment's membrane. Movement of soluble proteins from their endoplasmic reticulum to the Golgi apparatus occurs by vesicular transport. Those proteins that lack a sorting signal remain in the cytosol permanently. Trafficking places unique demands on membrane proteins for folding, translocation, and stability. Thus, much research is now being done to understand misfolded proteins, for example, as a cause of disease. Proteins facilitate transport across membranes by serving as receptors, enzymes, or transporters. Proteins act as recognition and binding units for substances moving in and out of the cell. They work as pores or transport channels for various electrically charged particles called ions or electrolytes, and specific carriers for amino acids and monosaccharides. And they work as specific enzymes that drive active pumps that promote concentration of certain ions, particularly your potassium, within the cell while keeping concentration of other ions, for example, sodium, below concentrations found in the extracellular environment. They work as cell surface markers, such as glycoproteins, that identify a cell to its neighbor, and they work as cell adhesion molecules, or proteins, that allow cells to hook together and form attachments to the cytoskeleton for maintaining cellular shape. And lastly, they work as catalysts for chemical reactions, for example, conversion of lactose to glucose. Membrane proteins are key components of energy transduction, converting chemical energy into electrical energy, or electrical energy into either mechanical energy or the synthesis of ATP. The interaction of plasma membrane proteins with lipids is complex and is the subject of much research. The role of proteins in the onset of and progression of disease is important because they govern communication between cells through enzymatic transport and recognition receptor functions in cellular physiology. So, protein regulation in a cell. Your proteostasis. Proteostasis is a state of cell balance of the processes of protein synthesis, folding, and degradation. Proteostasis is vital to cellular health. The cellular protein pool is in constant change or flux. The number of copies of a protein in a cell depends on how quickly it is made and how long it survives or is broken down. The system of protein homeostasis is adaptable and defined by proteostasis, network comprised of ribosomes, chaperones, and two protein breakdown systems, or proteolytic systems, lysosomes, and ubiquitin proteasome systems. The molecular chaperones, called heat shock proteins, are important and are so named because they increase after cells are exposed to elevated temperatures. The increase in heat shock proteins is part of the feedback system that responds to an increase in misfolded proteins and helps these proteins refold. These chaperones also facilitate transportation and ubiquitination, or tagging the protein with a small molecule, ubiquitin, that signals the protein to the proteasome for degradation. Together, all of these systems regulate protein homeostasis under a large variety of conditions, including nutrient supply variation, oxidative stress, cellular differentiation, temperature changes, heavy metal ion presence, and other stresses. Malfunction or failure of the proteostatics network is associated with human disease. So here you're taking a look at the protein homeostasis system and different outcomes. So again, here's a good spot to pause and take a look at how, for instance, you have a misfolding, folding, and aggregation. Proteases are enzymes that cause the breakdown of proteins. Certain proteases can be tethered to cell membranes. Proteases are involved in the physiologic regulation of essential processes by participating in a tightly orchestrated sequence of events, termed a proteolytic cascade. Four major proteolytic cascades with disease relevance are candidates for treatment modalities, including cell death or caspases-mediated apoptosis, the blood coagulation cascade, degrading membrane enzymes or matrix metalloproteinase cascade, and the complement cascade. Some proteases within the proteolytic cascade act as initiators. Others are involved in amplification and propagation and execution. Understanding the various steps involved is crucial for designing drug interventions. Dysregulation of proteases features prominently in human disease, including cancer, autoimmunity, and neurodegenerative disorders. The short chains of sugars or carbohydrates contained within the plasma membrane are generally bound to membrane proteins and lipids. Long polysaccharide chains attached to membrane proteins are called proteoglycans. All of the carbohydrates on the glycoproteins, proteoglycans, and glycolipids is located on the outside of the plasma membrane, and the carbohydrate coating is called the glycocalyx. The glycocalyx helps protect the cell from excuse me. The glycocalyx helps protect the cell from mechanical damage. Additionally, the layer of carbohydrates gives the cell a slimy surface that assists with mobility and other cells, like leukocytes, to squeeze through the narrow spaces. The functions of carbohydrates are more than protection and lubrication and include specific cell-to-cell recognition and adhesion. Intercellular recognition is a function of membrane. Oligosaccharides, for example, the transmembrane protein called lectins that bind to a particular oligosaccharide, recognize neutrophils at the site of a bacterial infection. This recognition allows the neutrophil to adhere to the blood vessel wall and migrate from the blood into the infected tissue to help eliminate the invading bacteria. So cellular receptors. Cellular receptors are protein molecules on the plasma membrane, in the cytoplasm, or in the nucleus that are capable of recognizing and binding with specific smaller molecules called ligands. The region of a protein that associates with a ligand is called its binding site. Hormones, for example, are ligands. Recognition and binding depends on the chemical configuration of the receptor and its smaller ligand, which must fit together somewhat like pieces of a jigsaw puzzle. Binding selectively to a protein receptor with high affinity to a ligand depends on formation of weak, non-covalent interactions, hydrogen bonds, electrostatic attractions, and van der Waals attractions, and favorably hydrophobic forces. Numerous receptors are found in most cells, and ligand binding to receptor activates or inhibits the receptor's associated signaling or biochemical pathway. So here you're taking a look at the ligand binds to the receptor. So you have your cellular receptors, the plasma membrane receptors for a ligand. Again, a good spot to pause so you can review that. Although the chemical nature of both ligands and the receptors to which they bind differ, receptors are classified on the basis of their location and function. Cellular type determines overall cellular function, but plasma membrane receptors determine which ligands a cell will bind with and how the cell will respond to binding with each. For example, the ability of a hormone or a neurotransmitter to stimulate a cell is regulated by the specificity and number of receptors present on the plasma membrane. Specific processes also control intracellular mechanisms. Hormone binding, for example, depends on special messenger molecules that regulate protein synthesis within the cell. Neurotransmitters also operate by causing special messengers to react with specific receptors. Receptors for different drugs are found on the plasma membrane, in the cytoplasm, and in the nucleus. Membrane receptors have been found for certain anesthetics, opiates, endorphins, encephalins, antibiotics, cancer chemotherapeutic agents, digitalis, and other drugs. Membrane receptors for endorphins, which are opiate-like peptides isolated from the pituitary gland, are found in large quantities in pain pathways of the nervous system. With binding, the endorphins change the cell's permeability to ions, increase the concentration of molecules that regulate intracellular protein synthesis, and initiate molecular events that modulate pain perception. Receptors for infectious microorganisms, or antigen receptors, bind bacteria, viruses, and parasites. Antigen receptors on white blood cells recognize and bind with antigenic microorganisms and activate the immune and inflammatory response. Cell-to-cell adhesion. Cells are small and squishy, not like bricks. They are enclosed only by a flimsy membrane, yet the cell depends on the integrity of this membrane for its survival. How can cells be combined together strongly with their membranes intact to form a muscle that can lift those textbooks? Plasma membranes not only serve as the outer boundaries of the cells, but also allow groups of cells to be held together robustly. And cell-to-cell adhesions to form tissues and organs. Once arranged, cells are held together by three different means: the extracellular matrix, cell adhesion molecules in the cell's plasma membrane, and specialized cell junctions. So the extracellular matrix and basement membrane. Cells can be bound together by attachment to one another through the extracellular matrix, which the cells secrete themselves. The extracellular matrix is an intricate meshwork of interstitial fibrous proteins embedded in a watery, gel-like substance composed of complex carbohydrates. A specialized type of extracellular matrix is called the basement membrane. This sheet of matrix molecules is very thin, tough, and flexible, lies beneath epithelial cells, and surrounds individual muscle cells, fat cells, and Schwann cells. The extracellular matrix is like glue. However, it does provide a pathway for diffusion of nutrients, wastes, and other water-soluble traffic between the blood and tissue cells. Overall, the matrix helps regulate cell growth, movement, and differentiation. Specifically, the major function of the extracellular matrix include mechanical support, control of cell proliferation, formation of scaffold for tissue regeneration, and establishment of tissue microenvironments. Interwoven within the matrix are three groups of macromolecules: fibrous structural proteins, including collagen and elastin, a diverse group of adhesive glycoproteins such as fibronectin, and proteoglycans and hyaluronic acid. So here you see the extracellular matrix and how cells stick together. Again, a good spot to pause to review how that works. And here you're taking a look at the three ways the basement membranes are organized. Again, a good spot to pause to review your biology. If you would like to read Box 1.1, here is a good spot to pause: the functions of the extracellular matrix. Collagen forms cable-like fibers or sheets that provide tensile strength or resistance to longitudinal stress. Collagen breakdown, such as occurs in osteoarthritis, destroys the fibrils and that give cartilage its tensile strength. Elastin is a rubber-like protein, fiber most abundant in tissue that must be capable of stretching and recoiling, such as the lungs. Fibronectin, a large glycoprotein, promotes cell adhesion and cell anchorage. Reduces amounts, Reduced amounts have been found in certain types of cancer cells. This allows cancer cells to travel or metastasize to other parts of the body. The extracellular matrix is secreted by fibroblasts, local cells that are present in the matrix. The matrix and the cells within it are known collectively as connective tissue because they connect cells together to form tissues and organs. Human connective tissue is enormously varied. They can be very hard and dense, like bone, flexible, like tendons, or the dermis of the skin, resilient and shock absorbing, like cartilage, or soft and transparent, like the jelly-like substance that fills the eye. In this In all these examples, the majority of the tissue is composed of extracellular matrix, and the cells that produce the matrix are scattered within it, like raisins in a pudding. The matrix is not just a passive scaffolding for cellular attachment, it also helps regulate the functions of the cell within it which it interacts. The matrix helps regulate cell growth, movement, and differentiation. Cell adhesion molecules, or CAMs. Cell adhesion molecules are cell surface proteins that bind the cell to an adjacent cell and to components of the extracellular matrix, or ECM. CAMs include four protein families: the integrins, the cadherins, the selectins, and immunoglobulin, the Ig superfamily. Integrins are a major class of receptors within the ECM and regulate cell ECM interactions with collagen, fibronectin, vitronectin, and fibrinogen. Cadherins are calcium-dependent glycoproteins and have unique pattern of tissue distribution, for example, the epithelial. Selectins are a family of proteins that bind certain carbohydrates, for example, mucins. The immunoglobulin superfamily CAMs, the IGSF CAMs, bind integrins or other IGSF CAMs. Cells, specialized cell junctions. Cells in direct physical contact with neighboring cells are often linked together at specialized regions of their plasma membranes, called cell junctions. Cell junctions are classified by their function: to hold cells together, forming a tight seal, so tight junctions; to provide strong mechanical attachments, your adherence junctions, desmosomes, hemidesmosomes; to provide a special type of chemical communication, so for example an inorganic ion and small water-soluble molecules to move from the cytosol from one cell to the cytosol of another cell, such as those causing an electrical wave, or gap junctions; and to maintain apicobasal polarity of individual epithelial cells, again, tight junctions. Overall, cell junctions make the epithelium leak-proof, allow communicating tunnels, and maintain cell polarity. Here is a good spot to hit pause if you need to review how different epithelial cells uh, communicate. Cell junctions can be classified as asymmetrical and symmetrical. Symmetrical junctions include tight junctions, the belt desmosome, desmosomes, and gap junctions, also called intercellular channel or communicating junctions. An asymmetrical junction is the hemidesmosome. Together, the zones between epithelial cells, typically consisting of sonula occludens, the sonula adherens, and the macula adherens, form the junctional complex. Desmosomes unite cells either by forming continuous bands or belts of epithelial sheets, or by developing button-like points of contact. Desmosomes also act as systems of braces to maintain structural stability. Tight junctions are barriers to diffusion, prevent the movement of substances through transport proteins in the plasma membrane, and prevent the leakage of small molecules between the plasma membranes of adjacent cells. Gap junctions are clusters of communicating tunnels, or connexons, that allow small ions of molecules to pass directly from the inside of one cell to the inside of another. Connexons are hemichannels that extend outward from each of the adjacent plasma membranes. The junctional complex is a highly permeable part of the plasma membrane. Its permeability is controlled by a process called gating, which depends on the concentrations of calcium ions in the cytoplasm. Increased cytoplasmic calcium concentration causes decreased permeability at the junctional complex. Gating is an important cellular defense mechanism because it enables uninjured cells to seal themselves off from injured neighbors. Damaged cells release calcium, which travel through the junctional complex and increases calcium levels in neighboring cells, causing damaging effects. The increased calcium concentration decreases the permeability of the junctional complexes of the neighboring cells, which form a relatively impermeable wall around the injured area. Cellular communication and signal transduction. Cells need to communicate with each other to maintain a stable internal environment or homeostasis, to regulate their growth and division and their development and organization into tissues, and to coordinate functions. Cells communicate by using hundreds of kinds of signal molecules. However, cells communicate in three main ways. They develop plasma membrane-bound signaling molecules, or receptors, that affect the cell itself and other cells in direct physical contact. They affect receptor proteins inside the target cell, and the signal molecule has to enter the cell to bind with them. Finally, they form protein channels that directly coordinate the activities of adjacent cells. Alterations in cellular communication affect disease onset and progression. In fact, if a cell is unable to perform gap junctional intercellular communication, it is hypothesized that normal growth control and cell differentiation are compromised, favoring cancerous tumor development. Secreted chemical signals involve communication at a distance. Primary modes of intercellular signaling are contact-dependent paracrine, hormonal, neurohormonal, and neurotransmitter. So here you're taking a look at cellular communication. And here you were taking a look at primary modes of chemical signaling. Again, hit pause if I'm going too quickly. Contact-dependent signaling requires cells to be in close membrane-to-membrane contact. In paracrine signaling, cells secrete local chemical mediators that are quickly absorbed, destroyed, or immobilized. Paracrine signaling usually involves different cell types. However, cells also may produce signals that they themselves respond to, which is called autocrine signaling. An example of this would be cancer cells use this form of signaling to stimulate their own survival and proliferation. Hormonal signaling involves specialized endocrine cells that secrete chemicals called hormones. Hormones are released by one set of cells and travel through the tissue and through the bloodstream to produce a response in other sets of cells. In neurohormonal signaling, hormones are released into the blood by neurosecretory neurons. Like endocrine cells, neurosecretory neurons release blood-borne chemical messengers, whereas ordinary neurons secrete short-range neurotransmitters into the small, discrete space. Neurons communicate directly with cells they innervate by releasing chemicals or neurotransmitters at specialized junctions called chemical synapses. The neurotransmitters diffuses across the synaptic cleft and acts on the postsynaptic target cell. Many of these same signaling molecules are receptors used in hormonal, neurohormonal, and paracrine signaling. The important differences lie in the speed and selectivity with which the signals are delivered to their targets. Plasma membrane receptors belong to one of these three classes that are defined by the signaling mechanisms used. The following table summarizes these classes of receptors. Again, a good spot to hit pause and review some of your anatomy. Signal transduction. Signal transduction involves incoming signals or instructions from extracellular chemical messengers. They are conveyed to the cell's interior for execution. Cells respond to external stimuli by activating a variety of signal transduction pathways, which are communication pathways or signaling cascades. Signals are passed between cells when a particular type of molecule is produced by one cell, the signaling cell, and received by another, the target cell, by means of a receptor protein that recognizes and responds specifically to the signal molecule. In turn, the signaling molecules activate a path of intracellular protein kinases that result in responses such as grow and divide, survive, or differentiate. If deprived of appropriate signals, most cells undergo a form of cell suicide, known as programmed cell death or apoptosis. So here you're taking a look at the schematic of signal transduction pathways. A good spot to hit pause if you need a quick review. Signal transduction pathways, or relay chains of intercellular signaling molecules, have several important functions. One, they physically transfer the signal from the place at which it is received to some other part of the cell where the response is expected. Two, they amplify the signal received, making it stronger. This is caused by a multiplying effect in the pathways. For example, binding of one ligand molecule to the receptor activates a number of adenylyl cyclase molecules. 3. They distribute the signal so that it influences several processes in parallel. At any step in the pathway, the signal can diverge and be relayed to several different intracellular targets, creating branches in the flow and causing a complex response. And 4. The signal can be modulated by other interfering factors prevailing inside or outside the cell. Two general responses from binding of the extracellular signaling messenger or first messenger to the membrane receptor occur, opening or closing specific channels in the membrane to regulate the movement of ions into or out of the cell, and transferring the signal to an intracellular messenger, or a second messenger, which in turn triggers a cascade of biochemical events within the cell. Your extracellular messengers and channel regulation. Membrane channels, or gates, can open and close depending on the circumstances of the first messenger. Opening and closing occurs because of conformational changes, shaping of the proteins that form the channels blocking the channel, or permitting passage through it. Channel opening and closing can be initiated in one of three ways: by binding of a ligand to a specific membrane receptor that is closely associated with a channel, by making changes in the electrical current in the plasma membrane, altering the flow of sodium and potassium, and by stretching or other mechanical deformation of the channel. The following figure summarizes ways by which extracellular messengers regulate channel function for the other two methods of controlling channels. So a good spot to pause and figure out how extracellular messengers regulate channel function. Nice little review here. Your second messengers. Many ligands cannot enter their target cells to cause the desired intracellular response. Instead, the first messengers, or ligands, issue orders by binding with receptors on the surface membrane, triggering a pass-it-on signal. Second messengers are generated in large number when the membrane-bound enzyme is activated, and they then rapidly diffuse away from their source, broadcasting the signal throughout the cell. Remember, most cell surface receptors, proteins, belong to one of three large classes: your ion channel linked receptors, your G protein linked receptors, or your enzyme linked receptors. So here's a good spot to hit pause and kind of review the first messenger and extracellular chemical messengers. The two major second messenger pathways are cyclic adenosine monophosphate, your cyclic AMP or cAMP, and calcium. In the cAMP pathway, binding of the ligand to its surface receptor eventually activates the enzyme adenylyl cyclase on the inner surface of the membrane. A membrane-bound middleman, a G protein, acts as an intermediary between the receptor and adenylyl cyclase. G proteins are named because they are bound to guanine nucleotides. Guanosine triphosphate, your GTP, or guanosine diphosphate, GDP. An unactivated G protein consists of a complex of alpha, beta, and gamma subunits, with a GDP molecule bound to the alpha subunit. The cAMP pathway with G proteins is summarized in the figure 1.24. Instead of CAMP, some cells use calcium as second messengers. In this pathway, binding of the first messenger to the surface receptor eventually leads, by means of G proteins, to activation of the enzyme phospholipase C, an enzyme protein effector that is bound to the inner side of the membrane. Figure 1.25 summarizes the calcium second messenger pathway. The cAMP and calcium pathways frequently overlap in triggering a specific cellular response. An example, your cAMP and calcium can influence each other. Calcium-activated calmodulin can regulate adenylyl cyclase and thus influence cAMP. Conversely, a cAMP-dependent kinase may phosphorylate and thereby change the activity of calcium channels or carriers. In some instances, both calcium and cAMP regulate the same intracellular protein. In a few cells, cyclic guanosine monophosphate serves as a second messenger similar to cAMP pathways. Another example, your cGMP is the signal transduction pathway involved in vision. Some cellular responses mediated by cAMP and phospholipase C are summarized in Table 1.3. Major types of receptors and signal transduction pathways are contained in Table 1.4. So now it's pause time again, and you can take a look at the first messenger and extracellular chemical messengers and the binding. The extracellular messenger and activation of the calcium second messenger system. And here's Table 1.3. You see some hormone-induced cell responses mediated by cAMP. The big ones here, for instance, is epinephrine, glucagon, ACTH, your antidiuretic hormone, acetylcholine, antigen, thrombin. And here's Table 1.4, major types of receptors and signaling transduction pathways. A large number of human disorders involve problematic signaling in cells. Cancer, for example, results from genetic mutations, leading to overactivity of proteins in signal relaying pathways that normally induce the cell to divide. Affected proteins cause cells to behave as if other cells were constantly telling them to reproduce, even when no such orders are sent. Signal blockers are already in use against tumors, leading us to cellular metabolism. All the chemical tasks of maintaining essential cellular function are referred to as cellular metabolism. The energy-using process of metabolism is called anabolism, and the energy-releasing process is known as catabolism. Metabolism provides the cell with the energy it needs to synthesize cellular structures. Dietary proteins, fats, and starches are hydrolyzed in the intestinal tract into amino acids, fatty acids, and glucose, respectively. These constituents are then absorbed, circulated, and taken up by the cell, where they may be used for various vital cellular processes, including the production of ATP. The process by which ATP is produced is one example of a series of reactions called a metabolic pathway. A metabolic pathway involves several intermediate steps whose end products are not always detectable. A key feature of cellular metabolism is the directing of biochemical reactions by protein catalysts, or enzymes. Most biochemical reactions in a pathway are catalyzed by a specific enzyme. Each enzyme has a high affinity for a substrate, a specific substance that is converted to a product of the reaction. So the role of adenosine triphosphate, best known as ATP, is its role as the universal fuel inside living cells. This fuel or energy drives biologic reaction necessary for cells to function. For a cell to function, it must be able to extract and use the chemical energy contained within the structure of organic molecules. When 1 mole of glucose is metabolically broken down into the presence of oxygen, into carbon dioxide, and water, 686 kilocalories of energy are released. In a test tube, this energy is released as heat. Because a cell cannot transform heat into work, chemical energy rather than heat is created by metabolism. The chemical energy lost by one molecule is transferred to the chemical structure of another molecule by an energy carrying or energy transferring molecule, such as ATP. The energy stored in ATP can be used in a variety of energy requiring reactions, and in the process is generally converted to adenosine diphosphate and inorganic phosphate. The energy available as a result of this reaction is about 7 kilocalories per mole of ATP. In addition to its use in synthesis of organic molecules, ATP is used by the cell for muscle contraction and active transport of molecules across cellular membranes. The function of ATP is not only to store energy, but also to transfer it from one molecule to another. Energy is stored by molecules of carbohydrate, lipid, and protein, which, when catabolized, transfer energy to ATP. Food and production of cellular energy. The process of catabolism of the proteins, lipids, and polysaccharides found in food can be divided into the following 3 phases. So if you look at the following 3 steps right here, you see the phases of catabolism. So a good spot to hit pause. So phase 1, your digestion. Large molecules are broken down into their smaller subunits, proteins into amino acids, polysaccharides into simple sugars, and fats into fatty acids and glycerol. These processes occur outside the cell by the action of secreted enzymes. Phase 2, your glycolysis and oxidation. The small molecules enter cells and are further broken down into the cytoplasm. Most of the sugars are converted into pyruvate. Pyruvate then enters mitochondria and is converted into the acetyl groups of acetyl coenzyme A. Acetyl CoA, like ATP, releases energy when it is hydrolyzed. The most important part of this phase 2 is the lysis or splitting of glucose, known as glycolysis. Glycolysis produces a net of 2 molecules of ATP per glucose molecule through the process of oxidation, or the removal and transfer of a pair of electrons. This process, often called oxidative cellular metabolism, involves 10 biochemical reactions. In reactions 1 through 5, glucose is converted to 2 3-carbon aldehyde compounds, which require energy in the form of ATP. The next 5 reaction converts the G3P molecule into pyruvate molecules and generate 4 molecules of ATP for each 2 molecules of G3P. In addition, 2 molecules of nicotinamide, adenine dinucleotide, are further oxidized to produce 4 more molecules of ATP. After subtracting 2 molecules of ATP to drive the reaction, the net yield is 6 ATP molecules for each molecule of glucose. So here you're taking a look at glucose. Good spot to hit pause and remember your biochemistry and chemistry and biology. All the steps to glycolysis. In phase 3, citric acid cycle or Krebs cycle, also known as the tricarboxylic acid cycle. Most of the ATP is generated during this final phase. It begins with the citric acid cycle and ends with oxidative phosphorylation. About two-thirds of the total oxidation of carbon compounds in most cells is accomplished during this phase. The major end products are carbon dioxide and two dinucleotides, reduced nicotinamide adenine dinucleotide and the reduced form of flavin adenine dinucleotide, which transfer their electrons into the electron transport chain. Oxidative phosphorylation. Oxidative phosphorylation occurs in the mitochondria and is the mechanism by which the energy produced from carbohydrates, fats, and proteins is transferred to ATP. During the breakdown of foods, many of the reactions involve the removal of electrons from various intermediates. These reactions generally require a coenzyme, such as nicotinamide adenine dinucleotide, to transfer the electrons and thus are called transfer reactions. Molecules of NAD and flavin adenine dinucleotide, or FAD, transfer electrons they have gained from the oxidation of substrates to molecular oxygen. The electrons from reduced NAD and FAD, NADH and FADH2 respectively, are transferred to a series of carrier molecules on the inner surface of the mitochondria with the release of hydrogen ions. Some carrier molecules are a group of brightly colored iron-containing proteins, known as cytochromes, that accept a pair of electrons. These electrons eventually combine with molecular oxygen. If oxygen is not available to the electron transport chain, ATP will not be formed by the pathway synthesis ATP. This process, called substrate phosphorylation or anaerobic glycolysis, is linked to the breakdown of carbohydrates. So here is what happens to pyruvate. Another good spot to hit pause. Because glycolysis occurs in the cytoplasm of the cell, it provides energy for cells that lack mitochondria. However, as noted, glycolysis also provides energy to the cell when oxygen delivery is insufficient or delayed. The reaction in anaerobic glycolysis involves the conversion of glucose to pyruvic acid with the simultaneous production of ATP. With the glycolysis of one molecule of glucose, two ATP molecules and two molecules of pyruvate are liberated. If oxygen is present, the two molecules of pyruvate move into the mitochondria, where they enter the citric acid cycle. If oxygen is absent, pyruvate is converted to lactic acid, which is released into the extracellular fluid. Elevated lactate level is indicative of tissue hypoxia, or low oxygen concentration. The conversion of pyruvic acid to lactic acid is reversible. Therefore, once oxygen is restored, lactic acid is quickly converted back to either pyruvic acid or glucose. The anaerobic generation of ATP from glucose through the reactions of glycolysis is not as efficient as the aerobic generation of ATP. The addition of an oxygen-requiring stage to the catabolic process provides cells with a much more powerful method for extracting energy from food molecules. Your membrane transport, the cellular intake and output. Cell survival and growth depends on the constant exchange of molecules with their environment. Cells continually import nutrients, fluids, and chemical messengers from the extracellular environment and expel metabolites or the products of metabolism, and end products of lysosomal digestion. Cells also must regulate ions in their cytosol and organelles. Simple diffusion across the lipid bilayer of the plasma membrane occurs for such important molecules as oxygen and carbon dioxide. However, the majority of molecules transfer depends on specialized membrane transport proteins that span the lipid bilayer and provide private thoroughfares for select molecules. Membrane transport proteins occur in many forms and are present in all cell membranes. Transport by membrane transport proteins is sometimes called mediated transport. Most of these transport proteins allow selective passage. For example, sodium but not potassium, or potassium but not sodium. Each type of cell membrane has its own transport proteins that determine which solute can pass into and out of the cell or organelle. The two main classes of membrane transport proteins are transporters and channels. These transport proteins differ in the type of solute, small particles of dissolved substances they transport. A transporter is specific, allowing only those ions that fit into the unique binding site on the protein. A transporter undergoes conformational changes to enable membrane transport. A channel, when open, forms a pore across the lipid bilayer that allows ions and selective polar organic molecules to diffuse across the membrane. Transport by a channel depends on the size and electrical charge of the molecule. Some channels are controlled by a gate mechanism that determines which solute can move into it. Ion channels are responsible for the electrical excitability of a nerve and muscle cells and play a critical role in the membrane potential. So here you're taking a look at the inorganic ion and a small polar organic molecule and can cross a cell membrane through either a transporter or a channel. The mechanism of membrane transport depend on the characteristics of the substance to be transported. In passive transport, water and small electrically uncharged molecules move easily through pores in the plasma membrane lipid bilayer. This process occurs naturally through the semi-permeable barrier. Molecules will easily flow downhill from a region of high concentration to a region of low concentration. This movement is called passive because it does not require expenditure of energy or driving force. It is driven by osmosis, hydrostatic pressure, and diffusion, all of which depends on the laws of physics and do not require life. Other molecules are too large to pass through pores or are ligands bound to receptors on the cell's plasma membrane. Some of these molecules are moved into and out of the cell by active transport, which does require life, biologic activity, and the cell's expenditure of metabolic energy. Unlike passive transport, active transport occurs across only living membranes that have to drive the flow uphill by coupling it to an energy source. Movement of a solute against its concentration gradient occurs by special types of transporters called pumps. These transporter pumps must harness an energy source to power the transport process. Energy can come through ATP hydrolysis, a transmembrane ion gradient, or sunlight. The best known is the sodium-potassium dependent adenosine triphosphatase pump, also known as ATPase. It continuously regulates the cell's volume by controlling leaks through pores or protein channels and maintaining the ionic concentration gradient needed for cellular excitation and membrane conductivity. The maintenance of intracellular potassium concentrations is required also for enzyme activity, including enzymes involved in protein synthesis. Large molecules, along with fluid, are transported by endocytosis and exocytosis. Receptor macromolecule complexes enter the cell by means of receptor-mediated endocytosis. So here we're taking a look at the pumps that carry out active transport in the three ways. So, a good spot to hit pause and review. Mediated transport systems can move solute molecules singly or two at a time. Two molecules can be moved simultaneously in one direction or in opposite directions, or in a single molecule can be moved in one direction. So here you're taking a look at mediated transport. Another good spot to hit pause and review if you're unfamiliar. Electrolytes as solutes. Body fluids are composed of two types of solutes: electrolytes, which are electrically charged and dissociate into constituent ions when placed into solution, and non-electrolytes, such as glucose, urea, and creatinine, which do not dissociate. Electrolytes account for approximately 95% of the solute molecules in body water. Electrolytes exhibit polarity by orienting themselves towards the positive or negative pole. Ions with a positive charge are known as cations and migrate toward the negative pole, or cathode, if an electrical current is passed through the electrolyte solution. Anions carry a negative charge and migrate toward the positive pole, or anode, in the presence of electrical current. Anions and cations are located in both the intracellular fluid and the extracellular fluid compartments, although the concentration of particular ions varies depending on their location. An example of this is sodium is the predominant extracellular cation, and potassium is the principal intracellular cation. The difference in ICF and ECF concentrations of these ions is important to the transmission of electrical impulses across the plasma membrane of the nerves and muscle cells. Electrolytes are measured in milliequivalents per liter or milligrams per deciliter. Milliequivalents per liter indicate the number of electrical charges per unit volume of fluid. The term milliequivalent thus indicates the chemical combining activity of an ion, which depends on the electrical charge, or valence, of its ions. In abbreviations, valence is indicated by the number of plus or minus signs. Monovalent ions, or ions with one charge, include sodium, chloride, and potassium. Divalent ions, which have two charges, include calcium and magnesium. One milliequivalent of any cation can combine chemically with one MEq of any anion. One monovalent anion will combine with one monovalent cation. Divalent ions combine more strongly than monovalent ions to maintain electrochemical balance. One divalent ion will combine with two monovalent ions. An example of this is calcium and 2-chloride, making calcium chloride. Passive transport, diffusion, filtration, and osmosis. Diffusion is the movement of a solute molecule from an area of greater solute concentration to an area of lesser solute concentration. The difference in concentration is known as the concentration gradient. Particles in a solution move randomly in any direction. If the concentration of particles in one part of the solution is greater than that in another part, the particles distribute themselves evenly throughout the solution. According to the same principle, if the concentration of particles is greater on one side of a permeable membrane than on the other side, the particles diffuse spontaneously from the area of greater concentration to the area of lesser concentration until equilibrium is reached. The higher the concentration on one side, the greater the diffusion rate. The overall effect of diffusion is the passive movement of particles down a concentration gradient. That is, from an area of high concentration to an area of low concentration. The diffusion rate is influenced by differences of electrical potential across the membrane. Because the pores in the lipid bilayer are often linked with calcium, other cations diffuse slowly because they are repelled by positive charges in the pores. The rate of diffusion of a substance depends also on its size and its lipid solubility. Usually, the smaller the molecule, the more soluble it is in oil, the more hydrophobic or nonpolar it is, and the more rapidly it will diffuse across the bilayer. Oxygen, carbon dioxide, and the steroid hormones are all examples of nonpolar molecules. Water-soluble substances such as sugars and inorganic ions diffuse very slowly, whereas uncharged lipophilic molecules such as fatty acids and steroids diffuse rapidly. Ions and other polar molecules generally diffuse across cellular membranes more slowly than lipid-soluble substances. So here you are taking a look at passive diffusion of solute molecules across the plasma membrane. Water readily diffuses through biologic membranes because water molecules are small and uncharged. Although the mechanism is not known with certainty, the dipolar structure of water allows it to cross rapidly through the regions of the bilayer containing the lipid head groups. Lipid head groups constitute the two outer regions of the lipid bilayer. Filtration, hydrostatic pressure. Filtration is the movement of water and solutes through a membrane because of a greater pushing pressure, or force, on one side of the membrane than that of the other side. Hydrostatic pressure is the mechanical force of water pushing against cellular membranes. In the vascular system, hydrostatic pressure is the blood pressure generated in vessels by the concentration or contraction of the heart. Blood reaching the capillary bed has a hydrostatic pressure of 25 to 30 milligrams of mercury, which is sufficient force to push water across the thin capillary membranes into the interstitial space. Hydrostatic pressure is partially balanced by osmotic pressure, whereby water moving out of the capillaries is partially balanced by osmotic forces that tends to pull water into the capillaries. Water that is not osmotically attracted back into the capillaries moves into the lymph system. So here we're taking a look at hydrostatic pressure and oncotic pressure in plasma. Osmosis is the movement of water down a concentration gradient that is across a semi-permeable membrane from a region of higher water concentration to a region of lower water concentration. For osmosis to occur, the membrane must be more permeable to water than to solutes, and the concentration of solutes must be greater so that water moves more easily. Osmosis is directly related to both hydrostatic pressure and solute concentration, but not to particle size or weight. For example, particles of the plasma protein albumin are small, but more concentrated in body fluids than the larger and heavier particles of globulin. Therefore, albumin exerts a greater osmotic force than globulin. Osmolality controls the distribution and movement of water between body compartments. The terms osmolality and osmolarity are often used interchangeably in reference to osmotic activity, but, they define different measurements. Osmolality is a measure of the number of milliosmoles per kilogram of water, or the concentration of molecules per weight of water. Osmolarity is a measure of number of milliosmoles per liter of solution, or the concentration of molecules per volume of solution. When solute is added to water, the volume is expanded and includes the original liter of water, plus the volume occupied by the solute particles. In measuring osmolarity, the volume of water is therefore reduced by an amount equal to the volume of added solute. In solutions that contain only dissociable substances, such as sodium and chloride, the difference between the two measurements is negligible. In considering all the different solutes in plasma, however, the difference between osmolality and osmolarity becomes more significant. In plasma, less of the plasma weight is water, and the overall concentration of particles is therefore greater. The osmolality will be greater than the osmolarity because of the smaller proportion of water. Osmolality is thus preferred measure of osmotic activity in clinical assessment of individuals. The normal osmolality of body fluids is 280 to 294. The osmolality of intracellular and extracellular fluid tends to equalize and therefore provide a measure of body fluid concentration and thus the body's hydration status. Hydration is also affected by the hydrostatic pressure, because the movement of water by osmosis can be opposed by an equal amount of hydrostatic pressure. The amount of hydrostatic pressure required to oppose the osmotic movement of water is called osmotic pressure of the solution. Factors that determine osmotic pressure are the type and thickness of the plasma membrane, the size of the molecules, the concentration of molecules, or the concentration gradient, and the solubility of molecules within the membrane. Examples of movement of water in relation to hydrostatic and osmotic force occur in the glomerulus of the kidney and the capillaries of the microcirculation. Effective osmolality is sustained osmotic activity and depends on the concentration of solutes remaining on one side of the permeable membrane. If the solutes penetrate the membrane and equilibrate with the solution on the other side of the membrane, the osmotic effects will be diminished or lost. An example of this is urea is the small solute that readily diffuses across cellular membranes. Solutions containing urea rapidly lose their effective osmolality because they rapidly equilibrate. Solutes too large to pass through the membrane thus sustain an effective osmolality, meaning that they enhance osmotic activity. Plasma proteins are examples of molecules that provide effective osmolality because they normally do not cross cellular membranes. Plasma proteins also influence osmolality because they have a negative charge. The principle by which the plasma protein charge influences osmolality is known as the Gibbs-Donen equilibrium, and it affects the distribution of ions across cellular membranes. Gibbs-Donen equilibrium occurs when fluid in one compartment contains small, diffusible ions such as sodium and chloride, together with large, non-diffusible charged particles such as plasma proteins. Because the body tends to maintain an electrical equilibrium, the non-diffusible protein molecules cause asymmetry in the distribution of small ions. Anions, such as chloride, are thus driven out of the cell or plasma, and cations, such as sodium, are attracted. The protein-containing compartment will maintain a state of electroneutrality, but the osmolality will be higher. The overall osmotic effect of colloids, such as plasma protein, is called the oncotic pressure, or colloid osmotic pressure. Tonicity describes the effective osmolality of a solution. Solutions then have relative degrees of tonicity. An isotonic solution has the same osmolality or concentration of particles as the ICF or ECF. Diarrhea, for example, is the loss of isomotic fluid from the gastrointestinal tract. As a result, ECF volume decreases, but there is no change in ECF osmolarity. Examples of isotonic solutions include 5% dextrose in water and normal saline solution. Hypotonic solution has a lower concentration and is thus more dilute than body fluids. Water is a hypotonic solution. Consequently, water is osmotically pulled into the cells, causing cells to swell or burst. A hypertonic solution has a concentration greater than 285 to 294. An example of a hypertonic solution is 3% saline solution. Water can be pulled out of the cells by a hypertonic solution so that the cell shrinks. The concept of tonicity is important when correcting water and solute imbalances by administering different types of replacement solutions. Active transport of sodium and potassium. The active transport system for sodium and potassium is found in virtually all mammalian cells. The sodium-potassium antiport system uses the direct energy of ATP to move these cations. The transporter protein is an enzyme, adenosine triphosphatase, or ATPase, has a requirement for sodium, potassium, and magnesium ions. The concentration of ATPase in plasma membranes is directly related to sodium-potassium transport activity. Roughly 60% to 70% of ATP synthesized by cells, especially muscle and nerve cells, is used to maintain the sodium-potassium transport system. Excitable tissue have a high concentration of sodium-potassium ATPase, as do other tissues that transport significant amounts of sodium. An example again is the kidneys and your salivary glands. For every ATP molecule hydrolyzed, 3 molecules of sodium are transported out of the cell, whereas only 2 molecules of potassium move into the cell. The process leads to an electrical potential and is called electrogenic, with the inside of the cell more negative than the outside. The exact mechanism for the transport of sodium and potassium across the membrane is uncertain. One proposal is that ATPase induces the transporter protein to undergo several conformational changes, causing sodium and potassium to move short distances. The conformational changes creates a lowering affinity for sodium and potassium to the ATPase transporter, resulting in the release of cations after transport. So here we're taking a look at the movement of sodium and potassium. The sarcoplasmic reticulum of the heart muscle and skeletal muscle has an ATP-dependent calcium active transport system that regulates the calcium levels in the cell's cytoplasm, which in turn regulates muscle contraction and relaxation cycles. The calcium transport system depends on ATPase activity and is similar to that of your sodium-potassium ATPase. The transport of sugars and amino acids across the plasma membrane depends on the simultaneous movement of sodium and sodium-dependent transport. Sodium-dependent symport occurs primarily in the plasma membrane of the epithelial cells of the kidney tubules and intestines. The transport of glucose is not directly dependent on the hydrolysis of ATP. However, the sodium gradient is ATP dependent, and thus ATP is indirectly involved in glucose transport. The epithelial cells that line the intestines depend on sodium to transport various amino acids. Similarly, the uptake of chloride by the small intestine depend on sodium symport and antiport mechanisms for secretion of calcium from the cell. Table 1.5 summarizes the major mechanisms of transport through the pores and protein transporters in the plasma membranes. Many disease states are caused or manifested by the loss of these membrane transport systems. So again, a good spot to hit pause to review the major transport systems in mammalian cells. Transport by vesicle formation. Endocytosis. Endocytosis is a cellular internalizing process where a section of the plasma membrane enfolds substances from outside the cell, invaginates, and separates from the plasma membrane, forming an endocytic vesicle that moves into the cell of the cell. Endocytosis mediates cellular uptake of receptor ligand complexes, nutrients and their carriers, extracellular matrix components, bacteria, viruses, cell debris, and sometimes even other cells. Endocytosis regulates the composition of the plasma membrane and responds to changes in the extracellular environment, counterbalances exocytosis, and maintains homeostasis. Here's a nice picture of endocytosis and exocytosis. Depending on the size of the cargoes and other factors, binding of the ligands on the cell surface activates appropriate intracellular mechanisms that then trigger changes in membrane shape to mediate their cell entry. Endocytosis can be subdivided into four different categories: clathrin-mediated endocytosis, caveolae-mediated endocytosis, macropinocytosis, and phagocytosis. Over time, however, these categories may change. So here's a multiple pathway of endocytosis. Always a good spot to hit pause if you need to review. Because most cells continually ingest fluid and solutes by pinocytosis, the term pinocytosis and endocytosis are often used interchangeably. Micropinocytosis is the taking up of specific macromolecules by invagination of the cell membrane, which is then pinched off, forming a small vesicle in the cytoplasm. In micropinocytosis, the vesicle containing fluids, solutes, or both fuses with lysosome, and lysosomal enzymes digest them for use by the cell. Macropinocytosis is when a large fluid-filled vesicle or macropinosome is pinched off from the cell membrane and brought into the interior of the cell. Macropinocytosis is an important pathway for antigen presentation by specialized antigen-presenting cells. In phagocytosis, the large molecular substances are engulfed by the plasma membrane and enter the cell so that they can be isolated and destroyed by lysosomal enzymes. Substances that are not degraded by lysosomes are isolated in residual bodies and released by the cell by exocytosis. Both pinocytosis and phagocytosis require metabolic energy and often involve binding of the substance with plasma membrane receptors before membrane invagination and fusion with lysosomes in the cell. Clathrin-mediated endocytosis, ligand binding to some plasma membrane receptors, leads to clustering, aggregation, and immobilization of the receptors in the specialized areas of the membrane called coated pits. The pits, which are coated with a complex of proteins and cytosolic bristle-like structures, deepen and enfold, internalizing ligand receptors complexes and forming clathrin-coated vesicles. This internalization process is not completely understood, called clathrin-mediated endocytosis, is rapid and enables the cell to ingest large amounts of specific ligands, for example low-density lipoprotein, growth factor, and antibodies, without ingesting large volumes of extracellular fluid. After the coated pinch off from the plasma membrane, they quickly shed their coat and fuse with an endosome. An endosome is a vesicle pinched off from the plasma membrane, from which their contents can be recycled to the plasma membrane or sent to lysosome for digestion. Additionally, clathrin is required for the internalization of pumps and transporters for ions and small nutrients to maintain homeostasis and synaptic transmission in neurons. The ingested material is processed by lysosomal enzymes inside the cell. So here you see the ligand internalization by means of a receptor-mediated endocytosis. Again, a good spot to hit pause and hit review. Caveolae-mediated endocytosis. The outer surface of a plasma membrane is dimpled with tiny flask-shaped pits called caveolae. Caveolae are also called microdomains. Caveolae are cholesterol and glycosphingolipid-rich microdomains, where the protein caveolin is thought to be involved in the several processes, including the transportation and endothelial cell function, membrane repair, uptake of various cell surface receptors, protection against lipotoxicity, and mechanotransduction. Caveolae are present in most mammalian cell types and are abundant in endothelial and smooth muscle cells, adipocytes, and fibroblasts. Caveolae-mediated endocytosis has been implicated in the endocytosis of simian virus 40, papillo, papillo, papillomavirus, oh my god, sorry, papillomavirus, and cholera. Additionally, evidence is accumulating that these microdomains are important in regulating endothelial cell function, mainly because they compartmentalize various signaling molecules. Clathrin and caveolin-independent endocytosis. Some proteins or small molecules enter cells by clathrin and caveolin-independent pathways. These routes are defined and classified by the requirements for dynamin during fission and the regulatory roles of guanosine triphosphatases. The mechanism by which internalization occurs is not known, but is thought to be dependent on cargo concentration and membrane curvature. Much new research in the past decade has shown extracellular vesicles as important mediators of intercellular communication involved in biologic signaling to mediate a diverse number of biologic processes. Extracellular vesicles play a key role in the regulation of normal physiology, such as stem cell number, tissue repair, immune surveillance, and blood coagulation, and in the pathology of many diseases. Extracellular vesicles have been tightly linked to carcinogenesis, the dissemination of viruses such as HIV-1, amyloid beta-derived peptides, and alpha-synuclein, and the spread of abnormal cell surface prion protein. A new advancement in cellular biology is the role of exosomes in understanding the biology of vesicles and their role in disease and therapeutic potential. Exosomes are small membrane vesicles of endocytic origin, containing lipid, protein, and RNA species in a single biologic unit. Exosomes, secreted by nearly all mammalian cell types, confer messages between cells, proximal and distal, by transporting cargo in the form of protein, nucleic acid, and lipids. They are present in the intracellular space and in the body fluids, including plasma, saliva, urine, pleural ascites, amniotic fluid, cerebrospinal fluid, colostrum, breast milk, and semen. Importantly, tumor-derived exosomes can prepare a favorable microenvironment at future metastatic sites and mediate non-random patterns of metastasis. Investigators have defined a specific group of integrin expressed on tumor-derived exosomes, distinct from tumor cells, which dictate exosome adhesion to specific cell types and ECM molecules in particular organs. Recent data show that actin, cytoskeleton regulatory protein cortactin, promotes exosome secretion. Thus, exosome may be important not only to predict metastatic propensity, but also to determine organ sites of future metastasis. So here is a nice spot to hit pause and review the exosomes of cellular function. Exocytosis. In eukaryotic cells, secretion of macromolecules almost always occurs by exocytosis. Exocytosis is the discharge or secretion of material from the intracellular vesicle at the cell surface. An example of this to secrete macromolecules of insulin across plasma membranes. Insulin-producing cells store and package insulin molecules in intracellular vesicles, which fuse with the plasma membrane and open to the extracellular space, or matrix, releasing the insulin. Not all secreted substances are secreted into the extracellular matrix. Some adhere to the plasma membrane and are thought to replace segments of the membrane lost through endocytosis or diffuse into the blood to nourish or signal other cells. Research findings suggest membrane lipids may be a regulator or of exocytosis. Understanding how exocyst complex is regulated is critical for diverse physiologic processes. The intact phospholipid phosphoinositide plays a critical role in most, if not all, cellular events associated with plasma membrane, including vesicle exocytosis, endocytosis, cell adhesion, phagocytosis, viral budding, enzyme activation, ion channel regulation, and cytokinesis. Exocytosis has two main functions: replacement of portions of the plasma membrane that has been removed by endocytosis, and release of molecules synthesized by the cells into the extracellular matrix. Movement of electrical impulses, the membrane potential. All body cells are electrically polarized, with the inside of the cell more negatively charged than the outside. The difference in electrical charge, or voltage, is known as the resting membrane potential, and is about -70 to -85 millivolts. The difference in voltage across the plasma membrane is a result of the difference in the ionic composition of ICF and ECF. Sodium ions have a greater concentration in the ECF, and potassium ions have a greater concentration in the ICF. The concentration difference is maintained by the active transport of sodium and potassium, which transports sodium outward and potassium inward. Because the resting plasma membrane is more permeable than to potassium than to sodium, potassium can diffuse easily from the area of higher concentration in the ICF to its area of lower concentration in the ECF. Because sodium and potassium are both cations, the net result is an excess of anions inside the cell, resulting in the resting membrane potential. So here is a good picture of a sodium-potassium pump and propagation of an action potential. Nerve and muscle cells are excitable and can change their resting membrane potential in response to electrochemical stimuli. Changes in resting membrane potential convey messages from cell to cell. When a nerve or muscle cell receives a stimulus that exceeds the membrane threshold value, there is a rapid change in the resting membrane potential, known as the action potential. The action potential carries signals along the nerve or muscle cell and convey information from one cell to another. When a resting cell is stimulated through voltage-regulated channels, the cell membrane becomes more permeable to sodium. There is a net movement of sodium into the cell, and the membrane potential decreases, or moves forward from a negative value to zero. This decrease is known as depolarization. The depolarized cell is more positively charged, and its polarity is neutralized. To generate an action potential and the resulting depolarization, a critical value known as the threshold potential must be reached. Generally, this occurs when a cell has depolarized by 15 to 20 millivolts. When the threshold is reached, the cell will continue to depolarize with no further stimulation. The sodium gates open and the sodium rushes into the cell, causing the membrane potential to reduce to zero and then become positive. The rapid reversal in polarity results in action potential. During repolarization, the negative polarity of the resting membrane potential is re-established. As the voltage-gated sodium channel begins to close, voltage-gated potassium channels open. Membrane permeability to sodium decreases, and potassium permeability increases with an outward movement of potassium ion. The sodium gates close, and with the outward movement of potassium, the membrane potential becomes more negative. The sodium-potassium pump then returns the membrane to the resting potential by pumping potassium back into the cell and the sodium out of the cell. During most of the action potential, the plasma membrane cannot respond to an additional stimulus. This time is known as the absolute refractory period and is related to changes in permeability to sodium. During the latter phase of the action potential, when permeability to potassium increases, a stronger than normal stimulus can evoke an action potential, known as the relative refractory period. When the membrane potential is more negative than normal, the cell is in a hyperpolarized state. A larger than normal stimulus is then required to reach the threshold potential and generate an action potential. When the membrane potential is more positive than normal, the cell is hypopolarized state, and a smaller than normal stimulus is required to reach the threshold potential. Changes in the intracellular and extracellular concentration of ions, or a change in membrane permeability, can cause these alterations in membrane excitability. Understanding the mechanism of ion movement and membrane excitability led to the discovery of calcium blocking agents for heart disease and migraines. Cellular reproduction, the cell cycle. The most fundamental task for the generation of life is cellular reproduction. The only way to make a new cell is to duplicate a cell that already exists. The continuity of life depends on constant rounds of cell growth and division. The cycle of repeated duplication and division is called the cell cycle. Humans must make millions of cells every second to survive. So that is pictures of the cell cycle. Reproduction of gametes, the sperm and egg cells, occurs through a process called meiosis, which we'll talk more about in Chapter 4. The reproduction or division of other body cells involves two sequential phases: mitosis, or nuclear division, and cytokinesis, or cytoplasmic division. These two phases occur in close succession, with cytokinesis beginning towards the end of mitosis. Before a cell can divide, however, it must double its mass and duplicate all its contents. Most of the work of preparing for division occurs during the growth phase, called interphase. The cell cycle drives the alternation between mitosis and interphase in all tissues with cellular turnover. Most of the early work on the cell cycle was limited to microscopic observation of mitosis and cytokinesis. Interphase was considered the resting stage of the cell. With recent technologic advances, a considerable amount has been learned about the interphase part of the cell cycle. During interphase, many important processes are taking place, as the cell produces DNA, RNA, protein, lipids, and other substances. And each pair of chromosomes make exact copies of themselves. The four designed phases of the cell cycle are the G1 phase, which is the period between the M phase and the start of DNA synthesis, the S phase, in which the DNA is synthesized in the cell nucleus, the G2 phase, in which RNA and protein synthesis occurs, the period between the completion of DNA synthesis, and the next phase, M, the M phase, which includes both nuclear and cytoplasmic division. So here we're taking a look at the four phases of the cell cycle, talking specifically about the phases of mitosis and cytokinesis. Your interphase, the G1, S, and G2 phase, are the longest phase of the cell cycle. During interphase, the chromatin consists of very long, slender rods that are jumbled together in the nucleus. Late in the interphase, strands of chromatin begin to coil, causing them to shorten and thicken. The M phase of the cell cycle, mitosis and cytokinesis, begin with prophase, the first appearance of chromosomes. As the phase proceeds, Each chromosome is seen in two identical halves, called chromatids, which lie together and are attached at some point by a spindle attachment site called the centromere. The nuclear membrane, which surrounds the nucleus, disappears. Spindle fibers are microtubules formed in the cytoplasm. Spindle fibers radiate from the two centrioles located at opposite poles of the cell. The role of the spindle fibers is to pull the chromosomes to opposite sides of the cell. During metaphase, the next phase of mitosis and cytokinesis, the spindle fibers begin to pull the centromeres of the chromosomes. The centromeres become aligned in the middle of the spindle, which is called the equatorial plate of the cell. In this stage, chromosomes are easiest to observe microscopically because they are highly condensed and arranged in a relatively organized fashion in the two-dimensional equatorial plate. Anaphase begins when the centromeres split and the sister chromatids are pulled apart. The spindle fibers shorten, causing the sister chromatids to be pulled, centromeres first, toward opposite sides of the cell. When the sister chromatids are separated, each is considered to be a chromosome. Thus, the cell has 92 chromosomes during this stage. By the end of anaphase, 46 chromosomes are lying at each side of the cell. Barring mitotic errors, each of the two groups of the 46 chromosomes is identical to the original 46 chromosomes present at the start of the cell cycle. During the telophase, the final stage, a new nuclear membrane is formed around each group of the 46 chromosomes. The spindle fibers disappear, and the chromosomes begin to uncoil. Cytokinesis causes the cytoplasm to divide into roughly equal parts during this phase. At the end of telophase, two identical diploid cells, called daughter cells, have been formed from the original cell. The cell cycle control system. Various features of the cell cycle differ from one cell type to another, including the length of time to complete certain functions. Most of the variability in the cell cycle length in the adult body occurs during the time the cell spends in the G1 or G0 phase of the cell cycle. This length of time is different from the brief time a cell takes to progress from the beginning of the S phase through mitosis, typically 12 to 24 hours. Cells are capable of disassembly of their cell cycle control system and can withdraw from the cycle to a non-dividing state called G0. The reversibility of the G0 state varies in different types of cells. For example, most of the neurons and skeletal muscle cells are in a terminally differentiated G0 state. With their cell cycle control system disassembled, the molecular regulatory switches become permanently turned off, and cell division rarely occurs. In eukaryotic cells, the basic organization of the cell cycle is mainly the same. The cell cycle control system triggers the essential events of the cell cycle. The system operates like a timer that triggers the constant sequence of events. Although early embryonic division are independent of feedback controls, in most cells the control system responds to feedback from the process it controls. Again, for example, if some malfunction occurs in the DNA synthesis, signals are sent to the control system to delay progression of the M phase. Such delays allow repair and prevent progression of cellular disasters. The cell cycle control system works because of a connected series of biochemical switches that either activate or inhibit cell events. Two classes of switches, or regulatory molecules that determine a cell's progress through the cell cycle, are cyclin-dependent kinases and cyclins. So here you're taking a look at the cell cycle control system. Cyclin-dependent kinases are protein kinases, and their function rise and fall as the cell moves through the cycle. The rise and fall leads to cyclical changes in phosphorylation of intracellular proteins that start or regulate the events of the cell cycle. Many enzymes and other proteins regulate the different CDKs, and the most important are proteins called cyclins. CDK activity is dependent on binding of cyclins. In another example, activation of the S phase, cyclin CDK complexes, initiates the S phase. Without cyclin, CDK is inactive. Binding of CDK inhibitors inactivates cyclin CDK complexes. Two other essential enzyme complexes that regulate the cell cycle control system are the APC-C and SCF, ubiquitin ligases, which promote ubiquitination and subsequent destruction of various proteins that modulate the cell cycle. That leads us to control of cell division and cell growth. Your mitogens, growth factor, and survival factors. The size of an organ depends on the total number of cells and cell size. Cell number depends on the total quantity of cell division and cell death. Organ size and body size are determined by three main processes: cell growth, cell division, and cell survival. These processes are tightly regulated by intracellular programs and extracellular signal molecules. The signal molecules that regulate growth, division, and survival are usually soluble proteins, protein bounds to cells, or components of the extracellular matrix. The molecules can be divided into three main classes: mitogens, growth factor, and survival factors. A mitogen is a substance that induces or stimulates mitosis. For a cell to proliferate, it must receive an extracellular signal, or mitogen, for other cells, usually neighboring cells. Mitogens relieve intracellular braking mechanisms that block processes through the cell cycle by triggering a wave of G1 SCDK activity. One of the first mitogens to be identified was platelet-derived growth factor. Importantly, when blood clots, platelets that exist in the clot are stimulated to release their contents of their secretory vesicles, such as the PDGF, liberated PDGF helps stimulate blood clotting at sites of tissue damage to prevent excessive bleeding. PDGF can stimulate many cell types, including smooth muscle cells, neurological cells, and fibroblasts. In addition to cell division, PDGF and other mitogens can stimulate cell growth, differentiation, migration, and survival. Mitogens interact with cell receptors to activate multiple intracellular signaling pathways. A major pathway through the GTPase is RAS, which leads to activation of mitogen-activated protein kinases cascade. Eventually, this cascade leads to the production of transcription regulatory proteins, including MYC. MYC is very important because it activates many genes involved in cell growth and is under investigation as a contributor to cancer cell stemness. Tumor appearing to be addicted to MYC, and the persistence of many human cancers requires sustained MYC activation. Activation of MYC leads to increase in expression of many delayed genes, including those that increase your G1 CDK activity, causing phosphorylation of a molecular on-off switch. The retinoblastoma susceptibility protein. In its unphosphorylated state, RB prevents cells from replicating by forming a tight, inactive complex with the transcription factor E2F. Phosphorylation of RB eliminates the brakes, and the liberated E2F proteins then activate other target genes, promoting cell cycle progression and cell replication. So here we're taking a look at growth factor and how it moves, the signaling cascade of RAS, MAPK, and MYC. Again, a good spot to hit pause. And here is a picture of RB, master regulator of cell cycle. Growth factor, also called cytokines, stimulate an increase in cell mass or cell growth by promoting the synthesis of proteins and other macromolecules and inhibiting their degradation. The following table includes examples of mitogens and growth factors. Cells that are starved of growth factors come to a halt after mitosis and enter the arrested or G0 state of the cell cycle. Survival factors promote cell survival by suppressing the type of programmed cell death called apoptosis. So here's another great spot to hit pause to know your different growth factors. See if I can put it all on one slide. There you go. Your DNA damage response blocks cell division. The DNA damage response occurs when DNA is damaged and several protein kinases are recruited to the site of damage and start a signaling pathway that stops the progression of the cell cycle or cell cycle arrest. Importantly, the cell cycle control system detects DNA damage and arrests the cycle at 2 points: at start, which prevents entry into the S phase, and at the G2M transition, stopping entry into mitosis. DNA damage initiates a signaling pathway by activating one of a pair of protein kinases, called ataxia telangiectases mutated, or ATM, and ataxia telangiectasia and RAD3, or ATR. So here we're taking a look at DNA damage response systems. Again, a good spot to hit pause if you're unfamiliar. And anytime you feel like I'm going too fast, hit that pause button. Tissues. The body is made up of 4 levels of organization: cells, tissues, organs, and systems. Cells of common structure and function are organized into tissues, of which there are 4 primary types: muscle, neural, epithelial, and connective. Tissue formation. To form tissue, cells must exhibit intercellular recognition and communication, adhesion, and memory. Specialized cells sense their environment through signals, such as growth factor from other cells. This type of communication ensures that new cells are produced only when and where they are required. Different cell types have different adhesion molecules in their plasma membranes, sticking selectively to other cells of the same type. They also can adhere to extracellular matrix components. Strength can occur because of the extracellular matrix and the strength of the cytoskeleton, with cell-to-cell adhesion to neighboring cells. Cells have memory because of specialized patterns of gene expression evoked by signals that acted during embryonic development. Memory allows cells to autonomously preserve their distinctive character and pass it on to their progeny. From generation, or morphogenesis, the cells undergo to assembly into tissue and organs with different shapes and sizes, is less well understood than the process of gene expression and induced signaling that results in cell type specialization. The underlying molecular mechanisms are complex. Fully specialized or terminally differentiated cells that are lost are generated from committed proliferating precursor cells, or transit amplifying cells, and they in turn have been derived from a smaller number of stem cells. Stem cells are cells with the potential to develop into many different cell types during early development and growth. A stem cell itself is not terminally differentiated. It can divide without limit, and when it divides, each daughter cell has a choice. The daughter cell can either remain a stem cell or move on a course that commits it to a terminal differentiation. 2 ways a stem cell can produce daughter cells with different fates are asymmetric division and independent choice, as shown in the following figure. Again, a good spot to hit pause if you're not familiar with stem cell and asymmetric division. In many tissues, stem cells serve as an internal repair and maintenance system, dividing indefinitely. These cells can maintain themselves over very long periods of time, called self-renewal, and can generate all the differentiated cell types of the tissue, or multipotency. This stem cell-driven tissue renewal is very evident in the epithelial lining of the intestine, stomach, blood cells, and skin, which is continuously exposed to environmental factors. Tissue renewal and repair, however, do not always depend on stem cells. An example of this is the enlargement and renewal of insulin-producing cells in the pancreas, occur by simply duplicating of existing insulin-producing cells. In the liver, differentiated hepatocytes can divide throughout life and significantly increase their division rate when needed. At the extreme opposite end of the spectrum, some tissues like the ear and the eye do not undergo any turnover and are not renewable. Once lost, they are forever lost. So different types of tissue are epithelial tissue. Epithelial tissue covers most internal and external surfaces of the body. Epithelial cells are closely joined and are attached to a basement membrane or lamina, which provides a supporting layer and separates the epithelium from underlying connective tissue. Because of its variety of location, epithelial tissue has several diverse functions, including protection, absorption, secretion, and excretion. Examples of this: the epidermis provides a protective barrier between the host and the outside environment, and the lining of the internal body organs helps absorb substances into the body, excrete waste products, and secrete substances into body cavities. Epithelial cell surfaces differ according to their location and function. Epithelial cells that line body cavities and blood vessels are smooth, whereas other epithelial cells have tiny cytoplasmic projections called microvilli on their free surfaces. Microvilli considerably increase the cell's surface area and are found on cells whose main function are absorption and secretion, such as the epithelial cells lining the digestive tract. Cilia, which are hair-like projections that propel mucus, pus, and dust particles out of the body, characterize cells lining the respiratory passage. Epithelial tissue is classified in two ways: according to the number and arrangement of cell layers, and according to their cell shape. Epithelium, that is formed by a single layer of cells, all of which are in the contact with the basement membrane, are called simple epithelium. Stratified epithelium has two or more layers of cells, and only the deepest layer is in contact with the basement membrane. Tissue that appears to consist of several cellular layers, but is actually a single layer with all cells contacting the basement membrane, is called pseudostratified epithelium. Three basic cell shapes are found in epithelium: squamous, cuboidal, and columnar. Squamous are flat and thin, cuboidal cells are high as they are wide and thus appear square in vertical section, and columnar cells are taller than they are wide and appear rectangular in vertical sections. Overall, classification of epithelial tissue, which takes into account both the number of cells, layers, and the cell shape, are summarized in Table E1.1 on Evolve. Connective tissue. Connective tissue varies considerably in structure and function, but is most common as the framework on which epithelial cells cluster to form organs. Other functions include binding various tissues and organs together, supporting them in their locations, and serving as storage sites for excess nutrients. In contrast to epithelial tissue, connective tissue is characterized by an abundant extracellular matrix that surrounds a few cells. The extracellular matrix is composed of ground substances and fibers. Ground substance is a homogeneous mass that varies in consistency from fluid to semi-solid gel. Fibers are produced by connective tissue cells found within the ground substance. The three types of fibers are collagenous, white, elastic, yellow, and reticular. Collagenous fibers are formed of bundles of smaller fibers appearing as wavy bands under the microscope. These fibers are composed of the protein collagen and are strong and inelastic. Elastic fibers are long, branching fibers composed of a protein called elastin that enables the fibers to return to their original length after stretching. Elastin occurs not only as fibers, but also as membranes, particularly the membranes of blood vessels. Reticular fibers are thin, short, branching fibers that form an inelastic network made from a collagen-like protein called reticulum. Reticular fibers form the internal framework, or stroma, to which the epithelial cells of glands are attached. They are found in loose connective tissue, generally in bone, marrow, and the parenchyma of the liver, spleen, and lymph nodes. Connective tissues are classified according to their consistency of the ground substance and the type of organization of the fibers within it. Table E 1.2 on Evolve summarizes characteristics of connective tissues. Muscle tissue is composed of long, thin cells or fibers called myocytes. Myocytes are highly contractile. The three types of muscle tissue are skeletal, cardiac, and smooth. Neural tissue. Neural tissue is composed of highly specialized cells called neurons, which receive and transmit electrical impulses very rapidly across junctions called synapses. Synapses are points of functional contact between neurons. At synapses, impulses pass from neuron to neuron or from a neuron to a muscle cell, while chemical messengers, called neurotransmitters, are released. The total number of neurons is fixed at birth, and replacement is impossible thereafter. Different types of neurons have specialized characteristics that depend on their distribution and function within the nervous system. All neurons, however, are composed of the following parts: a cell body, a single axon, and one or more dendrites. The cell body contains special cytoplasmic structures, as well as microtubules, actin filaments, Golgi complexes, lysosomes, and lipofuscin. The axon and dendrites can be very long. Generally, the axon conducts nerve impulses away from the cell body, and dendrites conduct nerve impulses toward the cell body. That here concludes our chapter. So, I thank you all for listening, for being part as always, and I'll see you at the next one. Thanks again for watching, and have a beautiful day.